Hello and welcome, dear students. Today we are going to discuss about the topic PTC Tasting Ability. The main objectives of today's lecture are to know the basics of PTC tasting, to discuss about the history of PTC tasting ability, to discuss about the PTC gene, to discuss the statistical methods and strategies for gene identification, to discuss the role of PTC in taste, to discuss the genetics of PTC, and finally to discuss the PTC taster stats and diseases. Dear students, let's start with the introduction first. Human population genetics aims to study the population in terms of genetic variation. The variation can be quantified by determining the gene frequencies of alleles at segregating loci which characterize one population and distinguish with another one. Tasting ability to phenyl thiocarbamide or PTC by an individual is considered as a useful and important tool to study the genetic diversity in human populations, also called a genetically controlled ability to taste phenyl thiocarbamide or PTC and a number of related substances, all of which have some antithyroid activity. It has the unusual property that it either tastes very bitter or is virtually tasteless depending on the genetic makeup of the taster. The ability to taste PTC is often treated as a dominant genetic trait, although inheritance and expression of this trait are somewhat more complex. About 70% of people can taste PTC, varying from a low of 58% of indigenous Australians and indigenous peoples of New Guyana to 98% for indigenous people of the Americas. One study has found that non-smokers and those not habituated to coffee or tea have a statistically higher percentage of tasting PTC than the general population. PTC does not occur in food, but related chemicals do, and food choice is related to a person's ability to taste PTC. Taste and smell affects food preferences and dietary habits, thereby directly influencing the eating behavior of an individual. As taste threshold increases with age, abnormality in taste function may contribute to poor dietary intake in the elderly. Bitter taste perception is a conserved chemical sense against the ingestion of naturally toxic substances in mammals. The experience of bitterness occurs after certain chemicals contact taste receptors located in cells on the surface of the tongue. Some investigators hypothesize that this sense provides information so that people do not ingest bitter tasting toxic chemicals. Studies of sensitivity to the bitter tasting antithyroid compound PTC have shown this to be an inherited trait, and non-taster stats has been linked to a variety of medical and health disorders. A high incidence of non-tasters have been reported among patients with nodular goiter, congenital atheriotic cretinism, and dental caries. Bitter perception generally occurs through bitter taste receptors located on the surface of taste cells of the tongue. These receptors are encoded by T2R genes that show 25 to 89% amino acid sequence identity between the 25 different members of this gene family. These differences presumably allow a wide variety of different chemical shapes, sizes, and functionalities to be bound by these receptors and perceived as bitter. In humans, responses to some bitter compounds show a bimodal distribution that distinguishes two phenotypes, tasters and non-tasters. The best studied example of these is the ability to taste PTC and other structurally related compounds. The importance of the ability to taste bitter chemical compounds like PTC was realized by Arthur Fox. Thereafter, Cinder showed that the inheritance of the ability to taste PTC was dependent on a single autosomal dominant gene. PTC is a bitter tasting, harmless chemical compound which is a member of a class of compounds known as thioureas. Researchers have identified a small region on chromosome 7q and harbors a gene that encodes a member of the TAS2R bitter taste receptor family. A major locus on the chromosome 7q35q36 and a secondary locus on chromosome 16p have been localized by genome scan for PTC taster gene. Tasters are those who taste the substance while non-tasters cannot taste at all. Tasters have the genotype dominant, that is TT, and the non-tasters have the genotype which is recessive, that is small tt. The ability to taste PTC is a dominant genetic trait, and the test to determine PTC sensitivity is one of the most commonly used genetic tests on humans. The strong genetic basis for sensitivity to PTC has been used as a tool to trace family lineage and population migration patterns. It was previously used in paternity testing before the advent of DNA markers. Studies reveal that human populations show a tremendous variation in the frequency of tasters, which range from 10% to 98%. Dear students, let's now discuss about the history of PTC tasting ability. In 1931, Arthur Fox was the first person who synthesized phenyl thiocarbamide or PTC. Some researchers reported a bitter taste while others, including Fox himself, experienced no such sensation of bitter taste. Further study of this phenomena by L.H. Cinder in 1931 led to the conclusion that the inability to taste phenyl thiocarbamide is a recessive trait. In 1932, Albert Blaxley conducted a large-scale study involving the inheritance of PTC tasting within families that concluded that PTC tasting sensitivity is very likely a complex Mendelian trait whose variance is overwhelmingly dependent on a single gene locus or position. However, it is likely that a few other genes have a smaller effect as well. In 1939, Fisher found that the genetic frequency of PTC tasting was the same in chimps and humans. This similarity suggests that whatever gene controls for PTC tasting must have some sort of a selective advantage in order to have either evolved and been maintained for millions of years. Since before humans and chimps diverged into separate species, since finding out that PTC tasting had an apparent naturally selective advantage, scientists began to hypothesize that this advantage was that the ability to taste natural chemicals similar to PTC helped human ancestors stay away from some toxic plants. Substances that resemble PTC today are in some vegetables from the cabbage family, that is Brassiaceae, such as broccoli and Brussels sprouts. In 1950s, William Boyd found evidence that the same gene that controls for the PTC tasting also controls for the tasting of a different compound that acts as an antithyroid drug similar to that found in cabbage plants. Despite all of this compelling evidence for PTC tasters to have a selective advantage over non-tasters, there was no explanation for the consistent proportions of non-taster in human and chimp populations. Until scientists discovered that subjects with a PTC non-taster phenotype were able to taste a different bitter compound that PTC tasters could not. This compound was juice from an Antidesma plant, and notably, this study found a perfectly inverse relationship between PTC and Antidesma tasting. However, in 2018, David Risso and colleagues expanded upon this relationship when they discovered a notable difference from previous studies. What they found was that there appeared to be a small population of people who could taste both PTC and antidesma as bitter. This finding could suggest a possible advantage for heterozygotes given their ability to taste both bitter compounds. Dear students, let's now discuss about the PTC gene. Soon after its discovery, geneticists determined that there is an inherited component that influences how we taste PTC. Today, we know that the ability to taste PTC or not is conveyed by a single gene that codes for a taste receptor on the tongue. The PTC gene TAS2R38 was discovered in 2003. There are two common forms or alleles of the PTC gene and at least 5 rare forms. One of the common forms is a tasting allele and the other is a non-tasting allele. Each allele codes for a bitter taste receptor protein with a slightly different shape. The shape of the receptor protein determines how strongly it can bind to PTC. Since all people have 2 copies of every gene, combinations of the bitter taste gene variants determine whether someone finds PTC intensely bitter, somewhat bitter, or without taste at all. Dear students, let's discuss about the mode of inheritance of PTC. Soon after the discovery of PTC sensory variation, several investigators reported that this trait is inherited and that the non-taster phenotype was transmitted as a simple Mendelian recessive trait. However, other studies have suggested that incomplete dominance, multiple alleles, or multiple genes fit the inheritance of the inability to taste PTC better than a simple Mendelian recessive model. Studies suggested that PTC taste sensitivity is controlled by a major locus with incomplete dominance as well as by a multifactorial component with significant residual heritability. Research studies produced results that supported a 2-locus model in which one locus controls PTC tasting and the other locus controls a more general taste ability. Thus, it became clear that PTC taste ability is not simply Mendelian in its transmission pattern, and rather than being a dichotomous trait, it displays a broad and continuous, albeit bimodal, distribution of taste sensitivity in the population. Dear students, let's now understand the statistical method and strategies for gene identification. Traditional parametric linkage methods may have low power to detect linkage for the PTC gene because of its high allele frequency. A significant fraction of the variation of PTC tasting in humans appears to be polygenic, and therefore parametric methods are a less desirable choice to map this trait. Affected sibling pair methods may be better suited to mapping the PTC locus. These methods do not rely on models that specify mode of inheritance, can detect multiple loci, and are capable of analyzing quantitative phenotypes such as threshold or suprathreshold measures of PTC stats. However, these methods are unproven because no gene has been cloned based solely on these methods alone. Because this taste polymorphism is found in primates, PTC tasting ability may be an ancient polymorphism, not a relatively new human mutation. Consequently, linkage methods such as homozygosity mapping, which is very powerful for mapping rare recessive disease, will have little power to map this trait. Dear students, let's discuss about the role of PTC in taste. There is a large body of evidence linking the ability of taste, thiourea compounds, and dietary habits. Much of this work has focused on 6-propyl-2-thiouracil or PROP, a compound related to PTC that has lower toxicity. heavy cigarette smokers are more likely to have high PTC and PROP thresholds that are relatively insensitive. In 1976, an inverse relationship between taster stats of PTC and for a bitter component of the fruit of the tree Antidesma buenos was discovered. Research on the implication still continues. Ability to taste PTC may be correlated with a dislike of plants in the genus Brassica, presumably due to chemical similarities. However, studies in Africa show a poor correlation between PTC tasting and dietary differences. Dear students, let's now understand the mechanism of bitter taste gene. Bitter taste grasp evolved as a key detection mechanism towards the ingestion of bioactive substances and is mediated using TAS2R gene family individuals invertebrates. The most notable recognized and amazing studied bitter substance is phenyl thiocarbamide or PTC, which is recognized through TAS2R38 and has a molecular structure related to that of glucosinolates contained in Brassica plants. The non-taster phenotypic polymorphism has been recognized in many primates. The genes and the food selections are intently connected. Taste and olfaction receptors may have advanced collectively at some stage in vertebrate evolution. The high frequency of genetic variations inside taste and olfactory receptors are special in human genome. Therefore, variations in these two perceptions can also be viewed as a landmark of human evolution. The sensory properties of foods, information, meals, preferences, and intake importantly deciding dietary and fitness stats. Variations in sensitivity to the bitter taste of phenyl thiocarbamide or PTC and different thiocarbamide containing compounds are properly documented. However, there is combined proof involving the association between bitter taste sensitivity, food preferences, and metabolic danger factors. Dear students, let's discuss about the genetics of PTC. Much of the variation in tasting of PTC is associated with polymorphism at the TAS2R38 taste receptor gene. In humans, there are 3 SNPs, single nucleotide polymorphisms, along the gene that may render its proteins unresponsive. There is conflicting evidence as to whether the inheritance of this trait is dominant or incompletely dominant. Any person with a single functional copy of this gene can make the protein and is sensitive to PTC. Some studies have shown that homozygous tasters experience a more intense bitterness than people that are heterozygous. Other studies have indicated that another gene may determine taste sensitivity. The frequency of PTC taster and non-taster alleles vary in different human populations. The widespread occurrence of non-taster alleles at intermediate frequencies much more common than recessive alleles, conferring genetic disease across many isolated populations suggested that this polymorphism may have been maintained through balancing selection. Chimpanzees and orangutans also vary in their ability to taste PTC, with the proportion of tasters and non-tasters similar to that in humans. The ability to taste PTC is an ancestral trait of hominids that has been independently lost in humans and chimpanzees through distinct mutations at TAS2R38 gene. Dear students, let's now talk about PTC heterozygotes are selectively advantageous over homozygotes. Based on the ubiquitous variation in PTC tasting ability in various populations and chimpanzees, it has been speculated that heterozygotes have a selective advantage over homozygotes. Otherwise, in the time elapsed since the creation of these alleles, selection or genetic drift or both would have eliminated one of the alleles from the population. The nature of the proposed selective advantage is unknown. One hypothesis, however, is that genotype at the PTC locus may influence food selection through its effects on bitter taste sensitivity, given that bitterness is associated with toxic compounds. PTC tasters may be more likely to avoid such toxic compounds, while PTC non-tasters might be willing to eat a broader variety of foods. These differences in food selection of bitter tasting foods may in turn influence metabolism and physiology. There is no single food category or food type, however, that is always preferred or avoided by PTC tasters. Dear students, let's discuss about the PTC taster stats and diseases. There have been reports of associations or lack of associations between PTC taste stats and diseases and traits not directly related to taste. These include diabetes, dental caries, eye diseases, thyroid disorders, schizophrenia, gastrointestinal ulcers, depression, mental function, growth variation, malignant tumors, and susceptibility to infectious diseases. The reports of association between diseases and taster stats may be due to chance associations. There are several instances of an initial report and one or more subsequent failures to replicate. In these cases, differences between studies in the characteristics of subjects or low statistical power may explain discordant results. Also, genetic association studies are prone to false positive results due to population stratification, which may be present in some but not all study populations. Finally, the associations may be genuine and could occur either because the taster locus is in linkage disequilibrium with other loci that predispose to a disease, or because the PTC locus has pleiotropic effects, or because the disease process changes PTC taster stats. Dear students, let's now discuss about the applications of PTC gene. Understanding of the multiple sequence variations in the PTC gene will allow examination of the details of the genetic basis of food preferences and the relationship between bitter taste sensitivities and possible health outcomes. Identification of the PTC gene and studies of its molecular evolution have revealed a number of different aspects that must be considered together in future studies of the genes responsible for inter-individuals differences in bitter taste sensitivity. First, unlike PTC, prop, and related compounds, the distribution of taste thresholds for other bitter compounds typically shows unimodal distribution. Second, although natural selection has acted to maintain both taster and non-taster alleles at the PTC locus, it is not clear whether this phenomena has occurred for other bitter taste receptor genes or not. There is a need to examine variation in the entire T2R gene family across populations. This will help human history in bitter taste perception and enable creation of comprehensive functional repertoires of each gene for studies of phenotype-genotype correlations. On a larger scale, the PTC gene may be illustrative of ancient genetic variation that has been proposed to underline common disease in modern populations. The PTC non-taster allele is common both because it is very old and because it appears to confer selective advantage, at least in the heterozygote state. This supports the common disease, common variant hypothesis and suggests that greater study of relatively older African populations could reap important benefits in the search for variation that underlies common diseases. Finally, PTC presents a unique opportunity in the field of bitter taste transduction. Having a known gene with a strong effect on phenotype in vivo provides many opportunities for studies of taste, physiology, biochemical function, and molecular structure elucidation in the human sense of taste. Dear students, let's conclude the lecture. PTC tasting ability is a simple genetic trait governed by a pair of alleles dominant T, that is capital T, for tasting, and recessive small t for non-tasting. Persons with genotypes capital T, the dominant genotype, and the capital T, small t, that is a heterozygous, are tasters, and the persons with a recessive genotype, that is small t, are non-tasters. There appears to be hormonal mediation of the tasting ability. However, because women are more often taste sensitive in this regard than are men, it has been suggested that PTC tasting may be related to the genetically determined level of tyrosine in the saliva. PTC tasting ability is not particularly useful since PTC does not occur in food, but some substances related to PTC do occur in food items. As for the utility of being able to taste PTC, it appears that non-tasters of PTC may have a higher than average rate of goiter, a disease of the thyroid gland, sometimes associated with a lack of iodine. Because PTC and related compounds contain iodine, there may be a selective advantage of some kind for tasters or non-tasters in different environments. It has also been suggested that tasters may have more food aversions than non-tasters, a disadvantage in situations of food scarcity. The chief reason for interest in tasting ability, however, is that the frequency of tasters varies from population to population. Dear students, it was all about today's lecture regarding PTC tasting ability. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.